Hello, you're welcome. Let's discuss population structure, one of the quality control measures during the Genome-wide association studies. What is population structure? In simple terms, it refers to a situation where subsets within your populations share more DNA or genomic variations with each other than the rest of the population. Sometimes this is because of geography or their breeding history. Now, in this plot here, you would see that there are some points or samples that are distributed over the map. At first glance, you don't see any clear groupings. But once we perform a population structure analysis, you can see that the same points now reorganize into distinct clusters. And usually, the clusters are linked to their geographic origins. And so in this example, you can see that they have clustered between America and the United Kingdom or Europe. So the question is, why do we care so much about the hidden subgroups in our population during a GWAS? First, structure can confound the GWAS results because the variants we find may only be chasing their ancestry rather than the traits or the phenotype you are interested in or you are working on. Second, structure can mask true associations because the model may not be able to detect or distinguish between the background variations in the true genetic variant. And finally, structure can reduce the statistical power of your model, and this could make it very difficult to detect the true variant. And so these scenarios gives a lot of problems for researchers and breeders. Now let's consider this scenario or example. So researchers thought they have found a single nucleotide polymorphism, here we call SNP-X, that controls drought tolerance. But later, they found that all the plants or the lines that were drought tolerant only came from the same region. Here we call it region A. And so it's likely that the shared ancestry between the points in region A would be the one driving the associations and not the causal variants. And so because of this, if you do not correct for population structure, you are likely only chasing a spurious association or a false positive. So now, this brings the question of how to correct for population structure. So there are several ways of doing this. One is building a kinship matrix, here represented by the funnel. So what this matrix will do is that it will quantify all those who are related or unrelated within your population. And so what we do is that we feed the matrix, or here the filter, with the raw data which includes the familial relatedness or the ancestry. And as a result, we get high quality genotypic data that is ready for our genome-wide association studies without any confounding ancestry or familial relatedness. And so in essence, the kinship or the kinship matrix or the rest of the other tools that detects the population structure, what they do is that they filter or soak up the familial relatedness or the ancestry within the population, so that whatever signals we get after the genome-wide association studies are likely the true genetic variants. And so in the upcoming videos, we are going to dive deeper into each of the methods that are described earlier. We are going to check how we can use principal component analysis to detect structure. And then we will dive deeper into structure and admixture models. And then we also have a look at the kinship in mixed models for GWAS analysis. If you enjoyed this video, kindly hit the subscription button and please press the bell so that you don't miss out in our next video or part 2 of this series. Kindly drop your questions or suggestions in the comment section. I will do well to address all of that in our next part. Thank you very much.